Hello students. In case of genetic engineering is a major breakthrough. And this breakthrough of this research has led to several other treatment which includes gene therapy for majority of this disease. I am Oviya from the Department of Biotechnology and I am here to talk about genetic engineering and the cloning vectors that are used in genetic engineering. So what is a vector? The vector is a major part in which the target gene is inserted, that is to obtain a recombinant DNA or a recombinant DNA plasmid. So this plasmid can be used in expression of a recombinant protein. So what is the characteristics of a vector? So it has to have the capability of replicating inside the host and it can autonomously replicate and it's smaller in size and easier to manipulate. So we have a major vector that is plasmid. So plasmids are circular and double-stranded DNA molecule. So here the plasmid can exist in many forms. That can be a circular, a supercoiled form, whereas it's a relaxed form is a covalently closed circular DNA. So what are these plasmids are? Where it is from? So naturally bacteria contains plasmids and it uses plasmids for major variety of purposes which includes antibiotic resistance, sugar fermentation, induction of plant tumor, host controlled modification and also heavy metal resistance. So naturally that are present in the environment and we are going to use these vectors in case of manipulating our own target gene of interest. So you have many types of plasmids which includes resistance plasmid, fertility plasmid, cold plasmid and degradative plasmid, where resistance plasmid is something where you have a particular resistance against for say antibiotic. So this resistant plasmid in turn which can even cause a treatment of bacterial infection very tedious because nowadays the antibiotic resistance among the bacteria is more common. It's because of this resistance plasmid. And you have other fertility plasmid which is used in conjugation purpose and cold plasmid that codes for coalescence and degradative plasmid which is an example toll from Pseudomonas. Other plasmid includes virulence. In case of virulence, a bacteria can become a pathogen. It can has the ability to cause disease because of these plasmids which can induce an infection in a host, especially a human. And natural plasmids that occurs both in eukaryotes and prokaryotes, co-leaven plasmid and artificial plasmid. These plasmids are the one we are talking about that we are going to use it for a recombinant DNA technology. Example PBR322, the first plasmid that was being identified and this can be used to clone a DNA. So these artificial plasmids, you have two types, both cloning and expression vector, where cloning is used to obtain a plasmid, a target genome can be modified by inserting a target gene sequence in the plasmid using this cloning vector, whereas the expression vector is used to express the protein of interest, that is the recombinant protein expression can be obtained. So cloning vectors are nothing but a simple vector where you can insert the target gene, obtain a recombinant plasmid and this can be transformed into a host. The host can be an E. coli, that is a bacterium, where generally commonly E. coli are used in case of transformation. Compared to plasmid, we have other vectors which includes phage, cosmid, bacterial artificial chromosome, yeast artificial chromosome and mammalian artificial chromosomes. Whereas to clone a large DNA segments, more than 1000 base pairs, generally people tend to go for other vectors compared to plasmid which includes BAC, YAC and mammalian artificial chromosome. In this case, you can use higher base pair, a bigger gene of interest and also it is used in post-translational modification. Whereas plasmid which is cloned and expressed in a bacteria cannot perform the host like how the eukaryote performs post-translational modification. Whereas these vectors, artificial vectors are used for obtaining a phosphorylated protein or a glycosylated protein. So it is an example plasmid PUC which is a simple derivative of PBR322 and bacteriophage. Other type of vector compared to a plasmid, it's a phage and phage is a vector where it can be both insertional and replacement vectors. In case of these vectors, it packs the target gene molecules into the lambda phage and the lambda phage can easily transduce those DNA targets into the vector. Where cosmids are plasmids that are packed into bacteriophage particles and these particles are in turn being constructed and it can use different genome fragments and the partially digested these fragments can be cloned in the vector. In case of backpack, the vector of choice is more than 100 kb. So it's used for cloning of higher genome sequence. Where BAC is a very simple plasmid and the DNA fragment size can range from 75 to 300 kb, and generally it is used in sequencing of genome of higher order organisms. Where YAC is an yeast expression vector and it's used cloning for very large DNA fragments and mapping of and physical mapping of more target sequences. Here we can see that expression vectors, that is to express a particular recombinant protein in a host, especially a bacterial or a mammalian host, to express the protein of interest we need an expression vector. Whereas it occurs using an inducible system where there is a particular promoter sequence in which the induction has to take place. Once the induction takes place, there is an activation, and based on the activation the recombinant DNA can be expressed and the protein can be seen. So the most commonly used host will be E. coli in case of bacteria, whereas in case of mammalian cell culture, the most commonly used is HEC293 and HeLa cells, where the recombinant expression can be both transient and permanent stable cells. So PCR, we know the enzymes that are used, DNA polymerases, stacked DNA polymerase, which I previously showed in a video. So this stacked DNA polymerase is used in PCR, that is polymerase chain reaction. The polymerase chain reaction is exactly used for amplifying a target gene, where amplification is very important in case of low expressing gene. We need to obtain more amplicons of the target gene sequence. Upon obtaining this gene sequence, we can get the insert, so the insert can be inserted into the target vector. This is the end. In the next video, we can see how these tools are used in obtaining a recombinant protein expression.